So this is the start of Unit 4. This is going to be 14.1 and 14.3, which is the cell theory and cell structure. For my honors kids, we plan on spending a little bit over 2 weeks on this, but not much longer than that. Some rough dates will be the 10th, you'll do your pre-test and begin vocab. 15th, vocab A will be due. 17th, vocab A quiz. 18th, vocab B due. 22nd, vocab B quiz. 24th, vocab C due. 25th, vocab C quiz. And the 31st will be your test and your projects due. Your project for this unit is an edible cell. The 31st is also Halloween, so kind of represents your Halloween party in a way. This unit is something you've covered before as well. We'll just go into a little bit more depth. So here we go. It was extremely nice in this unit and divided your vocab into 3 different parts. The first part is just basic terms for this unit. The second part is your cell parts, and the third part is more of your talking about what happens to the cell as it goes through different things. So we have the cell theory, prokaryote, eukaryote, nucleus, DNA, cell membrane, lipid bilayer, selectively permeable membrane. Part 2, nucleus, cell membrane, cell wall, ribosomes, smooth ER, rough ER, Golgi apparatus, chloroplast, mitochondria, lysosome, vacuole, cytoskeleton. And part 3, we have passive transport, diffusion, facilitate by diffusion, aquapore and osmosis, isotonic, hypertonic, hypotonic, active transport proteins, endocytosis, phagocytosis, pinocytosis, and exocytosis. For this unit, the honors are going to have to do a project. It is the edible cell project. I'm going to have it due on October 31st. With it being Halloween, we can't technically have a party, but with your edible projects due on Halloween, then we can call that our party. So what you have to do is construct an edible model of either a plant or an animal cell. Plan the ingredients for your creation and present your model to the class. The guidelines are that you must correctly identify if it is a plant or animal cell. Your model must be large enough to share with your whole class. You must include all the organelles that we learned about in class and they are listed below. If you want the full amount of extra credit, you should include at least 2 additional organelles. The organelles in your model should be at least slightly resembled to how the real organelle looks. You may need to do some additional research for this. Your textbook is a wonderful resource, as are online models. I would prefer you to label your model. However, with some kinds of food, it may not be really feasible to do this. Aren't sure what the word feasible means? Look it up. If you can't really label your model, you will need to be able to point to every organelle and tell me what it is and what it does from memory. No cheat sheets. To respect those students who have allergies, please do not use any nuts, peanut butter, or related items in your model. Oh, and no mushrooms, tomatoes, or bell peppers, just because I don't like them. And I'm the teacher, and I said so. Have fun and be creative. This is an edible project. There will not be an extended date. Instead, the student will have to do an alternate project per my choice. So the first part is 14.1, and this is the cell theory. There are 3 parts to the cell theory, and we will go through these during this time. This is the beginning of this unit, the history of cell biology. Cells are extremely small. How exactly small are they? Well, one human cell is smaller than a grain of sand. There are 5 prominent scientists that help introduce us to the cells that we know today. Not really good at pronouncing their names, but we'll just look at them. You have Robert Hooke, Anton van whatever, Swan, Virchow, and Sleeden, possibly. But those are the 5 scientists that brought us to where we are today with what we know about cells. The most obvious one that we think about when we hear cells is Robert Hooke. Robert Hooke, without him, we would not even know what the cells look like. Cells were not seen until the invention of the microscope, which he invented. In 1965, he observed the first cells from a slice of cork and called them cellulae, which in Latin stood for small rooms. There were 4 other scientists who were prominent in the discovery of the cell. The next one is Anton. He was the first one who saw a single-cell organism in pond water and observed cells in blood and semen. By the way, what is semen? This is a term that we will learn about later on, but you should become familiar with it now. Semen is a mixture of sperm and seminal fluids. Matthias Sleeden found out that all plants are made of cells. Theodor Swan found out that all animals are made of cells. And lastly, Rudolf Virchow found out that all cells come from pre-existing cells. After the work from all these 5 scientists was put together, they created what is known as the cell theory. The cell theory consists of all living things are made up of cells. Cells are the basic units of structure and function in living things, and new cells are produced from existing cells. Make sure that you remember those 3 things. It is very important. I guarantee you it will be on the EOC, it will be on your test, and you will definitely come in contact with it later on in life if you were to go into science. This is the big part of the unit. This is 14.3, and this is the cell structure and function. Here's where you will learn all the parts of both plant, animal, and bacteria cells, and will be responsible to know what each part does and what its job or niche is in the cell. All living organisms on Earth are made up of microscopic structures called cells. There are many different types of cells. Some organisms are unicellular, while other organisms, including humans, are multicellular. Cells generally share a similar structure. Most cells contain smaller structures called organelles. Organelles are groups of complex molecules that function like organs of a cell. Like the organs of the body, organelles of a cell perform different functions. This is known as the levels of organization. It consists of organ systems, which are made up of organs, which are made up of tissues, which are made up of cells. Examples of systems include the circulatory system, digestive system, or nervous system. Then we have the organs, which make up these systems, which are your lungs, stomach, heart, brain, and many more. Tissues are what make up these, which are like your muscles, and then the cells make up everything. Since this is a video, it cannot go exactly as to how my notes go. But as I go through this in class, you're going to be responsible for filling in the picture, which is already filled in on here, of this cell, which is an animal cell, and of the plant cell, which is the next picture. Also, as we go through it in class, you're going to be responsible for filling in that chart as I go through the notes. You're going to place a P, A, or B for whether if it's found in a plant cell, animal cell, or both. So make sure if you weren't here and you're trying to do this at home or whatever, that you do catch up on that. If it's not there, it will be marked wrong. But I'm going to go through the parts. You also are responsible of drawing what the basic structure looks like in your chart. The first part we have is the cytoplasm. This is the clear, gelatinous material found inside cells that supports and holds cellular organelles. It is the site of many cellular processes. The cytoplasm contains a cytoskeleton, which helps to maintain the cell's shape and structure. This is found in both animal and plant cells. The next part we have is the cell membrane. The cell membrane is also found in both plant and animal cells. It is a selectively permeable lipid bilayer that acts as a boundary layer around the cytoplasm, thus separating cells from their outside environments. In addition to being able to recognize chemical signals, the cell membrane is selectively permeable to chemicals and controls which molecules enter and leave the cell. Nutrients first enter the cell through the cell membrane. The cell wall is a secretion of the cell membrane that is found in plants, fungi, bacteria, and most protists. It provides protection from physical injury and provides structural support. It is not found in animal cells. The chloroplast is the food producer in a plant cell. During a process known as photosynthesis, chloroplasts absorb light energy from the sun and produce food in the form of glucose sugar from carbon dioxide and water. You'll learn more about photosynthesis in Unit 5 when we talk about plants. Chromatin is a collection of DNA and proteins that contains the hereditary information of an organism. The chromatin is loosely packed during interphase to allow for easy access for gene expression. Prior to cell division, the chromatin condenses to form chromosomes. We'll learn about this one in Unit 6, I believe, when we talk about mitosis and meiosis. The cilium is a tiny hair-like structure composed of microtubules that beats back and forth in order to allow the cell to move. Cilia are usually found in large numbers and can also aid in feeding by sweeping food towards the cell. Cells that line the digestive and respiratory systems of animals also have cilia that help with the movement of materials. The endoplasmic reticulum is found in both plant and animal cells. It transports proteins within the cells. The endoplasmic reticulum is also the location of lipid synthesis on the smooth side, and proteins are synthesized on the ribosome-studded rough ER. The flagellum is a tail-like structure composed of microtubules that is used by cells for movement. Plant and animal flagella move back and forth in order to propel the cell forward. Bacterial flagella, on the other hand, move in a corkscrew motion. The Golgi apparatus is also found in both plant and animal cells. It processes cellular waste from lysosomes as well as other materials and transports them out of the cell. In addition to playing an important role in waste disposal, the Golgi apparatus also processes, sorts, and modifies proteins in cells. The lysosome is a membrane-bound organelle found in animal cells that contains enzymes specialized to break down ingested material, secretions, and waste. These waste and other materials may be processed and transported out of the cell by the Golgi apparatus. The microfilament is a major component of a cell's cytoskeleton. They function in determining cellular shape as well as aiding in cellular movement. They are important for the formation of many different cellular projections that can allow a cell to move through its environment. Microtubules have two different components. They are components of the cilia and flagella as well as the cytoskeleton of a cell. Microtubules are hollow rods that help give a cell its shape and can also aid the cell in locomotion and the transport of materials through the cell. The mitochondria produce ATP, the body's primary energy source, through aerobic cellular respiration. They are only found in plant cells. The nuclear envelope is a double lipid bilayer that encloses the genetic material of a cell. It separates the nucleus of the cell from the cytoplasm. There are nuclear pores within the nuclear envelope that allow for the exchange of materials between the nucleus and the cytoplasm. The nuclear envelope is sometimes called the nuclear membrane. The nucleus is found both in plant and animal cells. It is the brain of the cell. It contains the nucleolus and the cell's DNA. The nucleolus is found within the nucleus of a cell and is responsible for synthesizing ribosomes and ribosomal RNA.